Hello बच्चों, today we are going to learn the second chapter of your exploration textbook, Cell, the Building Block of Life. In this chapter, we will be studying about the structure and functions of cell. What are the components of the cell called cellular organelles? What are their functions? What are their structures? How do cells divide? That means how do cells reproduce 1 to 2, 2 to 4, 4 to 8 like that. So first we start with level of organization. What is the first topic? Level of organization. See, your body is a huge factory, but the basic building blocks are the cells. You might have seen the construction of a building. The construction of a huge building starts with a single brick. The single brick, once they are clubbed together, the bricks they are joined together, it is forming the building. So just like that, our body, based on the complexity of the body, contains millions and trillions of cells. So the starting material or basic building block is the cell, both functionally and structurally. If you are checking the functions, the starting one, the basic one is cell. If you are checking the structure, also the starting or the basic one is cell. So cells are the basic units. Several cells together form the tissues. We call them tissues. So these cells will be having similar functions or structure, and we call them, when they are together, we call them tissues. Now, second level of organization, that is when these tissues which are there and they form a particular organ. Next one, several organs together form an organ system, just like the digestive system. Digestive system means it is not only the stomach and intestine. The digestive system starts from our mouth till anus, it spreads. So the entire organs which are there in this digestive system work unanimously for a single cause. For a single reason they are working, digestion, isn't it? So that's why it is called a system, digestive system. So several such kind of organ systems are there. And several such kind of organ systems together form the body of the organism. See, when we look at our body, we have digestive system, circulatory system, respiratory system, excretory system, reproductive system, several this kind of organ systems are there. So this kind of arrangement or organization is called level of organization, which start from a single cell till our body. Okay, next, how to study cells? See, cells are very small. We cannot see them using our naked eye. For that purpose, we need a sophisticated device called microscope. Micro means very small, isn't it? So this is a device or equipment which is used to see the smaller objects. And microscope, you studied about the function, structure of microscope in your previous year, class 8. Which will be having two different lenses. One is called objective, one is called eyepiece. Eyepiece means where we keep our eye to watch, and objective means where we keep our object, isn't it? So here we have a clarified picture of the microscope. Eyepiece you can see, objective lens you can see, stage on which we mount the slide, we keep the slide, and mirror, below that you can see a mirror to reflect the light. A handle is there, an adjustment knob that you rotate and adjust and get a clarified picture. Okay, so these are the main important points of a microscope. Using this microscope, you will be able to see the image of the object clearly. Okay, so another term which is introduced in your book is limit of resolution. It is the limit of your eyesight. and the value is 0.1 millimeter. It is told that when you have put two dots on the paper and you are watching these two dots from a distance. If the dots are closer together, very closer together, and if the distance between these two dots are smaller than 0.1 millimeter, we will not be able to see them separately. We will get a feeling that there is only one dot. They are together. There is only one dot. Such kind of feeling will be it is not the limit of that paper, it is the limit of our eye vision, and it is called limit of resolution. So in order to see the dots clearly, we should keep that paper at 25 centimeter away from our eyes. That is called the near point. Okay, near point. Clear vision is possible in that distance, 25 centimeter away from our eyes, and the distance between the 2 points should be more than 0.1 millimeter, so that we will be able to see them clearly as 2. Now, the next important point is an activity of the cell. So this activity gives you some important points about your cell membrane. Cell membrane, the universal feature of the cell. The cell membrane means it is a boundary, outermost boundary. And cell membrane is the protective layer which can be seen in both plant cells and animal cells. It's a membrane is a thin boundary that surrounds a cell and protects its contents. It defines the individuality of a cell. That means it is the limiting area. It defines the individuality of a cell. It is the boundary of a cell. It separates a cell from another cell and is also called plasma membrane. Very important. The other name of cell membrane is plasma membrane. The cell membrane is selectively permeable, which means it allows some substances to pass through while blocking others. Selectively permeable. Suppose you are going to your school, the gatekeeper, the watchman will allow those who are carrying an ID card. Selectively permeable, isn't it? Selectively allowing people to get inside and go outside. Just like that, our cell membrane, our plasma membrane is also selectively permeable. It will just allow some of the components to get inside and move out. Here is an activity to learn osmosis, and after that we will learn diffusion also. These are the two different processes in which the movement of particles can be seen. For osmosis activity, we need to take two equal sized potato slices. One potato, we will be keeping it beaker A and the other one in beaker B. Beaker A contains plain water and beaker B contains salt water. You can also take sugar water. No problem at all. We need to add some kind of salt or sugar into the water. Now, after some time, we will observe the changes happening to both the potato slices, and we will be taking out the potato slice and observe what is the difference between the two in shape. The first potato, which is put in beaker A, we can see that it is the shape of that potato has changed. It is now a little bit swollen. It is enlarged. It became bigger in size. And the second one, which is put in beaker B, will reduce in size, that it will shrink. So that process which is responsible for this change in shape is called osmosis. See, in plain water, there is plenty of water molecules when we compare it with potato. So the water molecules will move from its higher concentration area to lower concentration area through a semi-permeable membrane. That is what we call osmosis. And in the second case, the potato contains more water molecules than when we compare it with the salt water or sugar water. So obviously water molecules from its higher concentration, that is from inside the potato, will move outside to the water, to the salt water through a semi-permeable membrane. So here the direction is reversed. It is from the potato to the salt water, water is moving because we can see more number of water molecules in potato through a semi-permeable membrane. So that kind of process in which the solvent molecules are moving from higher concentration area to their lower concentration area through a semi-permeable membrane is called osmosis. You can see the definition here. Water moves from a high to low concentration across a semi-permeable membrane is called osmosis. No energy is needed. Because it's a spontaneous reaction. Now we can see what is diffusion. Diffusion means movement of particles from their higher concentration to their lower concentration without a semi-permeable membrane. Diffusion takes place in solid, liquid and gas. See, the best example is when you lighten an agarbatti, you will be able to smell the agarbatti from faraway places. That is due to the diffusion. The solid particles, smelly particles are taken or diffused into the air. Now we can see the difference between osmosis and diffusion. Diffusion takes place in liquid, solid or gas. Osmosis limited to liquid only. No need of a membrane, but in case of osmosis, a semi-permeable membrane is needed. Diffusion is which takes place in all directions, but osmosis is unidirectional. That means in one single direction only. So these are the differences between diffusion and osmosis. Now we will be meeting in the next video dealing about the structure of cell membrane in detail. Thank you so much.